Hello everyone, welcome to the session on metabolism. We'll be continuing with the oxidation of amino acids, which is part of the metabolism of amino acids. The first type which we in detail looked into was the deamination, the oxidative deamination and hydrolytic deamination. This next we have is transamination, transdeamination and decarboxylation. Okay, so first we'll see transamination. Transamination, it is a reversible transfer of alpha amino group from an amino acid to a keto acid. Okay, this keto acid, how it is formed? It is formed by deamination of another amino acid. Okay, so they are all like a reversible process. Now here you can see it involves the interconversion of a pair of amino acids and a pair of keto acids. What does it mean? Here we can start with this amino acid. Okay, this amino acid and this keto acid. These two substrates are undergoing transamination, during which the amino group from this amino acid, it will get transferred to the keto acid. As a result of which, what will happen is this amino acid, it will become its corresponding keto acid, and the keto acid which received the amino group will get converted into the corresponding amino acid. Okay, so there is a pair of amino acid and keto acid which when they undergo the transamination, it will result in another pair of keto acid and amino acid. Okay, the whole process, the whole reaction, it is catalyzed by transaminases, or it is otherwise known as aminotransferases. And this enzyme, it requires the presence of coenzyme pyridoxyl phosphate. Okay, now here you can see it is both deamination with respect to this amino acid and amination with respect to this keto acid. Okay, so it is a combined deamination and amination process which results in transamination. Transamination, it is very important with respect to the interconversion of an amino acid in metabolic processes. Okay, as already mentioned, it occurs with the help of amino acid specific transaminases. That is, for each set of amino acid, keto acid, there is a transaminases. Okay, now it occurs in all amino acids that process except for lysine, threonine, proline and hydroxyproline. Okay, and here there is no removal of ammonia, that is free ammonia is not liberated. Okay, now if you look into the salient features of transamination, we can see that the transamination, as already mentioned, it is a reversible process. It brings about the interconversion of amino acids in metabolic processes. It also channels the excess amino acids to energy generation. For example, when there are excess of amino acids, it can be used for the synthesis of certain products like ketoglutaric acid or pyruvic acid or oxaloacetic acid, etc. And these can then take part in the Krebs cycle and later be used for the synthesis of ATP. So in such way, the transamination process, it can channel the excess amino acids to generate energy. The transamination does not involve the release of free ammonia. It only involves intermolecular transfer of amino group. And most, as already mentioned, the lysine, except for lysine, proline, threonine, hydroxyproline, all amino acids take part in transamination process. And the most commonest amino group acceptors, they are alpha ketoglutaric acid or ketoglutarate, which when receives amino group becomes glutamate. Okay, then pyruvic acid, which when receives amino group, it will become alanine. Oxaloacetate, it is a keto acid of aspartic acid. Okay, so these are the major amino group acceptors. So transfer of amino group to these acceptors will result in the formation of the amino acids which are given in the brackets. Okay, now you can see here these are two major examples. Okay, the amino group of alanine and aspartic acid, it may get transferred to alpha ketoglutaric acid and results in the formation of these two. Okay, so aspartate is the amino acid and this is the keto acid, ketoglutarate. So this is the acceptor. So amino group from aspartic acid, it gets transferred to alpha ketoglutaric acid. It will result in the formation of oxaloacetic acid, which is a keto acid, and glutamate, which is the amino acid of the ketoglutarate. Okay, here also you can see alanine, it gets deaminated to produce pyruvic acid, while alpha ketoglutarate becomes aminated to become glutamate. These two are the reactions are catalyzed by aspartate aminotransferase and alanine aminotransferase enzyme. Okay, the transamination is a reversible reaction which is catalyzed by the aminotransferases or transaminases. And these enzymes, it is present in liver, pancreas, kidney, heart, and brain. And you can see the transamination process occurring in all these organs. Okay. Now you can see here, this is the transamination. Okay, the substrate amino acid. Okay, this is a step. This is a figure which shows the steps involved in transamination process. Okay, now here we have a substrate amino acid and a substrate keto acid. Okay, these two are the ones which are going into play. Right, this is the one, substrate amino acid and substrate keto acid. Now, how does it happen? Okay, the transamination, it involves two steps. Right, first one, it is transfer of amino group from the donor amino acid. This is the donor amino acid. Okay, transfer of amino group from the donor amino acid to pyridoxal phosphate, which is the coenzyme. Okay, so the amino group get transferred to pyridoxal phosphate. So this amino acid, it undergo deamination. As a result, it will turn into keto acid. Okay, when the amino group get transferred to pyridoxal phosphate, it will become pyridoxamine phosphate. Okay, so first the donor amino acid, it undergo deamination to produce keto acid, and the amino group released, it is taken up by the pyridoxal phosphate. This pyridoxal phosphate, as a result of amination, it becomes pyridoxamine phosphate. Okay, now the second step is, so what we have is pyridoxamine phosphate. Okay, now second step is transfer of this amino group from the pyridoxamine phosphate to keto acid. It is the amino acceptor, the acceptor amino acid, the keto acid. Okay, so this amino group, it will get transferred to the keto acid. What will happen? It will become the product amino acid, a new amino acid. And during this process, what happens? The keto acid will turn into amino acid. It releases pyridoxal phosphate free. Now, what all are the free compounds now? We have the product keto acid and product amino acid with pyridoxal phosphate free. I hope this is clear. Okay, now next is transdeamination. Transdeamination, it is a coupled reaction in which transamination, it is followed by oxidative deamination. Okay, and it is a major route of amino acid deamination. The net conversion of alpha amino group to ammonia, it requires the action of glutamate transaminase and glutamate dehydrogenase. So glutamate transaminase is involved in the transamination process, and glutamate dehydrogenase is involved in the oxidative deamination process. Okay, and the reaction occurs in both liver and kidney, in the mitochondria as well as in the cytoplasmic part. ATP and GTP are the allosteric inhibitors, while ADP and GDP activate the enzyme. Okay, now it is a reversible reaction, as already mentioned, and it usually functions in amino acid catabolism and amino acid anabolism. Okay, these two processes, you can see that transdeamination occurring. In amino acid catabolism, that is cutting down, the transmits nitrogen from glutamate to urea in urea cycle. So glutamate, the amino acid, is being cleaved to release the ammonia, so that it can be used for the urea synthesis. In anabolism, what happens? Amination of ketoglutaric acid. It is a keto acid, and as a result, what happens? Amination, it will result in the formation of glutamate. Okay, so it is a transdeamination process. So it involves catabolism, where deamination is taking place. Okay, and then followed by this one. Okay, so this is a step we can see. So transdeamination, it occurs in two steps. First step is transamination. Here you can see the transfer of amino group from alpha amino acid to a keto acid. Okay, alpha amino, what happens is, there is a amino acid and a keto acid involved in the reaction. Okay, amino group from alpha amino acid will get transferred to keto acid. What happens as a result? The keto acid will become an amino acid. What happens to the amino acid? It will become converted to the keto acid. Okay, and the whole reaction is catalyzed by transaminase enzyme. So a new amino acid and a keto acid are being produced. But here there is no involvement of ammonia. Okay, now second step is oxidative deamination. Here over here, this is the second step. Okay, here this undergoes oxidation, that is NAD to NADH2, and deamination. Here ammonia is released.